So cloning animals is where we were talking about yesterday, and the question on our review today was how do we do that? Well, we take an egg and we extract its nuclear material out, its genetic material out. So it's a cell that's missing its nucleus. Then we take a somatic cell. And a somatic cell is what? Where do we find somatic cells? Every cell in your body is a somatic cell, unless it's a sex cell. If it's not a sperm or an egg, it's a somatic cell. Okay, so we take one of those. I've read that with some of the animals, their preference was to take cells from the digestive tract. For some reason they think are healthier cells to use for this. I'm not sure how they came up with that information, but that's what I read that they would do. So they will take that healthy cell that has a complete set of chromosomes, so it's 2N, right? Complete set of chromosomes, 2N. And we'll take that cell, extract out its nuclear material, and we'll inject it into the ovum, the egg, and now it's 2N, just like it got fertilized, right? Went from haploid to diploid, 2N, when it got fertilized. And it will immediately start to grow and divide and grow into another organism, whatever I'm trying to clone. So we're trying to make a twin. Now, the only thing about this twin are usually twins are very similar in age. Like I remember asking you guys how when you were born, how many minutes apart, and you said it was what, 1 minute apart. But usually twins are usually within minutes apart. There are exceptions to that. I went to school with a set of twins up in Montana, and they were 5 months apart. Mom was pregnant, and then 5 months later became pregnant again, which that usually doesn't happen. You know, usually once you're pregnant, you can only be, until you deliver, you're not going to have any more. But in this case was an exception. She became pregnant again, but it's 5 months behind, a lot smaller. So when twin number 1 is born, there still is another baby inside the womb. Just real tiny. Yeah, right? And then 5 months later, yep, she was born. I don't know. Can't remember. It was, one was a boy and one was a girl, but I can't remember which one was born first. So, you know, like we've talked about in genetics, there's always exceptions. We learned in the first chapter what happens normally, and then now we've been talking about things that when it doesn't act normally. So now when they did the first mammal, which was the sheep, Dolly, they used somatic cells like it says here, but one of the things they discovered is that Dolly acted like she was much older than other sheep should be of her age. In other words, how long she's been since she's been born. And what we discovered, and she actually died early. What we discovered is, or at least is that, to this point, we think it has to do with when your mom and your dad, both when they were making their sex cells, remember it makes an exact copy of the parents? Well, in the processing of forming this new chromosome, there's something on the end of it called a telomere. And the telomere is long at first. And then every time it goes through a cell division, it somehow shortens. I don't know exactly how that works, but it actually gets shorter. And that basically, you know, how long is left on the end of the chromosome kind of determines how old you are. And so Dolly got a chromosome of a sheep that was, I forget how many years old, And so even though when she became mature, she was already, her chromosomes were already this old. And so she ended up dying much younger. So the question is, if we wanted to clone somebody who's 70 years old, if we ever were able to do that, their chromosomes are going to be like they're 70 years old. You don't have a long life left. So if you wanted to clone, you're going to have to figure out somehow when these new chromosomes are made, or we do it this way, that we can extend all the telomeres to make them like they are when you're brand new, made from a sperm and an egg. So I don't know that they've even figured out exactly how to do that. It's one thing to know what the problem might be. It's another thing to figure out, okay, then how are we going to work around that problem? But they began with an enucleated ovum. Enucleated mean that the nucleus, the ovum we had to take its nucleus out before we could put another one in. It doesn't have two, just has one from a somatic cell that is a complete set. Okay, here's just a description of that process, how they did it. They took obviously an egg out of a female sheep called a ewe, and did this process, and then put it back in, and Dolly was born. Sometimes. Oh, somebody gave me this. It needs to be what's. That's from last hour. Maybe my battery's dead. Okay, so I guess we'll have to do it the other way. Now, in human cloning, we've already talked about this a little bit. Identical twins are just natural clones. This is cloning that God's done. We talked about this a little bit yesterday. There's actually 3 things that make you who you are. What were the 3 again? The environment is one of them. The majority of what you look like is determined by the what? Yeah, we have a different soul. And then the third one is what? Your genes, okay? So your genes is, we get to see what your genes look like, right? The environment, we see only how those genes have been influenced by your environment. We don't know what you would look like if you were in a different environment. We don't know that. And then the soul, the spirit aspect of you that makes you who you are. And we talked about that yesterday, that Malin and Joella don't share a soul. Can you think about all kind of complications that don't seem to be consistent with the Bible, if that were true? What if you want to accept Jesus as your Savior, but Joella doesn't want to? That doesn't work that way, does it? So each has their own soul. Now, the real question is, should we clone humans? But another question that goes up, why should we? If we say we should, but why? Why would we? Well, what are some reasons that actually some people are pushing for cloning? What do you think? What are they looking for? So in other words, we could clone you. Let's say you have this disease of, let's say, your liver, okay? And maybe let's pick your heart instead, because the liver might be a little bit different, because you can actually donate part of your liver, and then your body will actually grow that part back. That's probably the only organ in your body that you can do that with. But let's say your heart. Okay, so you have a baby, it grows up, and then we take the baby's heart and put it into you so you can live longer. What's the problem with that?
Baby dies.
Yeah, the baby dies, right? You know, what makes you more important than this baby? You know, so, but there's some people that are pushing for that, you know, so you're basically cloning people so you can have spare parts. But they, you know, that's not the way God looks at you. You're not somebody's spare parts, are you? And the only time we're interested in your spare parts are when?
When you're dead.
When you
die.
When you die, you know, you can donate your organs, right? A lot of people donate their organs, and their organ actually goes on to, like heart is the one that you see most often. I think there was just something in the last week on the news where one family, their child died when it was, you know, still very young, and gave it to another child whose heart was born with a heart defect, and then they got to meet each other actually quite a few years later, and you know, quite often the parents of the child that died, you know, would like to hear their child's heart beating inside of this other individual whose life, you know, goes on. So yeah, we do that, but you know, we don't usually take organs from somebody until they're done with them. Now, we do that sometimes, like my friend whose son had kidney, like kidney cancer, I think it was. Well, at least something wrong with his kidneys. So they had he had to be on dialysis, and so my friend ended up donating one of his kidneys, which is okay because you have two. Okay, you can get by on one, you know. And so he gave one of his kidneys to his son, but whatever caused the disease, the genetic part that caused that disease, he still had. So it ended up affecting his donated kidney. So he needed to have a second kidney transplant, and then it got complicated. Then his mother. No, I have this backwards. His mother gave him a kidney, and it turned out the reason why he had this is because his mother has the genes for this disease. And so she gave him this kidney that would eventually become diseased. And so then he needed to have another kidney, and then his dad actually gives him his kidney. But mom, in the process, then her kidneys start failing. The one that she had left started failing. So then she had to get a kidney donated from her sister. But I think they figured out the sister didn't have that gene. So, I mean, it's kind of a complicated process, you know. But we figured out, to be able to do such things. Okay, now, genetic engineering is the next thing here. It's the manipulation of an organ's genome, in other words, your genetic makeup, by methods other than normal reproduction. We're going to adjust the genetic makeup of an organism without involving sperm and egg. And when we do that, one of the ways that we can do this is we can modify the DNA in one organism by getting some DNA or genetic material from a second organism. Quentin has what? What does Quentin have? He has diabetes, right? And he has to have insulin because his body doesn't make enough insulin. And so, since it doesn't have enough insulin, since he doesn't have enough insulin, he has to get insulin made for him then he has a little device that will add insulin that he's missing. Well, when they first started doing that with people who had diabetes, they discovered that some of the people that were getting the insulin, they were extracting insulin from, I thought it was sheep, but Quentin said he thought it was from pigs. One of those two. But they were allergic to that insulin. So, I mean, you're trying to fix one thing and now you made another problem. And so we've had to come up with a way of getting insulin for those people. So what we have done, and it'll talk about this here as we go forward, oops, too fast. We've extracted DNA or genetic material from someone who makes normal insulin. And we have put it into a bacteria's DNA, genetic material. So now we have a bacteria that already does bacteria stuff, but because of this genetic code that we've borrowed from a human, notice that we said it comes from another organism, we've we've taken some of that code and put it into a bacteria, and this bacteria now makes insulin, in addition to all the other bacteria stuff that bacteria do. And then we harvest that insulin, and I think that's the kind that Quentin has. He uses that kind of insulin. So we've taken genetic material from one organism and put it into another one so that that new organism will make the things that the original one couldn't. But we have to get good DNA, right? From somebody who's making insulin correctly. Does anybody happen to know what part of the body makes insulin? Starts with a P. Your pancreas. And there's actually a special part of the pancreas called the islets of Langerhans where actually the insulin's made. So it's not the whole pancreas, just a part of it. And so that's where the problem is, but we need somebody that has good genes for making insulin, and we're going to snip a piece out of that. And they talk about using restriction enzymes up here that we figured out a way. I don't know, it's always amazing that they figured out these things, where they can use an enzyme and it'll actually snip out of the DNA molecule the pieces that happen to do with, like in this case, making insulin. And then we take that piece and insert it into another organism's DNA. So now it has a new set of new set of genes that it never had before to make what it needs. In this case, it makes insulin. Now, how are you going to get that from one organism to the other organism? We talked about how we can snip it out, but how do you get it from there over to the other one? Well, one of the ways that we use is a vector. Now, a vector is an agent that carries genes from one organism to another. Viruses kind of do that. When a virus attacks one of your cells, it puts its genetic code inside of you. So if we could have a way of getting genetic material inside of this virus so that when it attacks the cell, it will put its genetic material into this cell. And so one way is actually using viruses. In other words, we're using viruses for a good purpose instead of making you sick. But we're using a virus to actually carry it to the new organism. And it also talks about plasmids in here, where that is, some DNA material that we're going to combine with another organism's material. And a plasmid, it's circular. It says it's a circular piece of bacterial DNA. So get it into the the DNA in the bacteria is a circle and you're going to put a piece in it. So it's not, you have to understand what organism you're taking it from and how we're going to, how does it match up with the pieces that are in, like bacteria, for instance. Now, genetic engineering, an organism that contains genes from more than one species. So when we took the DNA from one organism that made good insulin and put it into a bacteria, we've done that, haven't we? We've created a transgenic organism. Wasn't what God created. We've actually, in effect, made a new organism that has genetic material from two different organisms. So that's what we mean by transgenic organism. Okay, last topic we wanted to start today, we probably won't be able to finish that, is stem cells. Okay? Now remember we talked about this, that your cells, right at the very beginning, after the egg was fertilized and it started to divide, those cells are capable of becoming every kind of cell in your body. Okay? At first, there's nothing different about them. They're all alike. They're not like any cells you have in your body. In other words, looks-wise or function-wise. It just and at some point in the future, after this mass of cells is getting larger and larger, they will be like, these group of cells will start to become different. Like this group of cells will eventually, let's say, grow your arm and all the different parts that make up your arm. The bones, the muscles, the skins, all that kind of stuff. Or another part, it's going to, differentiate and become your leg. Or maybe a part of the cells, kind of like in the middle, may become all the organs that are inside of your body cavity. At one end, there are going to be a group of cells that are going to end up forming your head, and your brain, your eyes, your nose, your mouth, ears, all that is going to come out of those. So they have to become different at some point along the way. So, but stem cells are not differentiated yet. And they hold promise because they can potentially become other things. They are characterized by their potency. Now, what do we mean by that? One kind is totipotent, is a word that they refer to stem cells that are capable of becoming every type of cell in your body. In other words, if they differentiate, they'll eventually could become whatever. So if we put these stem cells, let's say in the brain, maybe they might develop into brain cells. Now, the process of them becoming different, is a process that we have to understand. What stimulates them to start becoming different? Is it just being near another cell, or is it the substances produced by that cell will cause their neighbors to start to become different? What? So there's some stem cells that can become any kind of cell in your body. There are other kinds of stem cells that we call pluripotent, and they can become many types of cells, but just not all the kinds. They're limited in how many different possibilities. So there's different kinds of stem cells out there now, where do we get the stem cells often from? Well, often from embryos, human embryos. How do we harvest these human embryos? What do you think the method has been to this point? Or not limited to this point, but was the main way. From what? A woman who's had an abortion. Okay, and depending how far along she is, they can harvest You know, these cells have started to differentiate already here, and these are the cells that are going to become different already. And we're going to take those out, and we're going to use them as stem cells. And we use, so the problem with that is, what does the Bible say about you?
That He knew us before we were formed in the womb.
Okay? God knew who we were even before we were in the womb. Last hour, I was able to use my cell phone. The only problem is my cell phone is now occupied. And so I'll have to read these out of, as our pastor calls this, an analog text. So we'll look at Jeremiah 13:16, the verses that Laura is mentioning. It says, "For you created my inmost being, you knit me together in my mother's womb." So inside of your mom, you were being put together. "I praise you because I'm fearfully and wonderfully made. Your works are wonderful. I know that full well. My frame," in other words, my body structure, was not hidden from you. In other words, while you were inside your mom, nobody could see you, but you were not hidden from God. When I was made in a secret place, so inside it's a secret place, nobody can look in there, even though we have figured out ways to take a peek. A doctor can take a peek. "And when I was woven together in the depths of the earth, your eyes saw my unformed body." So the whole time he's talking about you as a person, and you weren't even fully formed.